cook it, and then get out the food. Right? We don't want to do so many operations because more the operation that you do, more expensive and more difficult it becomes. So if I have, let us say, two steps, one is removal of lignin, one step, and then conversion of cellulose into sugar in the second step, and then conversion of sugar into ethanol in the third step. Can I do all in one vessel? I put everything, I close it, do some magic, so everything is, what I get out of that reactor is only ethanol. Right? So here is develop the process. At least maybe the first step need to be separate, but the second step where you hydrolyze and convert the hydrolyzed product into sugar, can we make them as a single step? People are looking at that because the more steps that you have, the more difficult. So we need to have the enzyme which can work under those conditions, and also the yeast or microorganism which can convert that sugar, hydrolyzed sugar, into ethanol. So develop consolidated process enzyme production. I'm sorry, you're not able to see that one. So the idea is to compact. If you have a large number of steps which are there, if I go back to this slide, you can see that there are a number of steps are there. You get this raw material, one step, second step, third step. But if I can combine at least these two in one unit operation, then it becomes much more economical to develop that process. But of course, as I said, that the another area where intellectual property rights or new ideas are being developed in cellulose material is not one cellulose. That means we have cellulose as well as hemicellulose. Cellulose is multiple glucose molecule joined together, hemicellulose multiple xylose molecule joined together. Saccharomyces cerevisiae can ferment only glucose, it cannot ferment your xylose. But then if I have 30% of the sugar is in the form of xylose, then I can't throw it out, it's a waste. So if I can have a microorganism which can work upon both the substrate and convert them into sugar, right, then I have a viable process. I'm using 100% of that water. So Pune University, Dr. Clarkson was developed a multiple sugar interesting Saccharomyces by genetic modification, right? And inserting a gene which can ferment the xylose also simultaneously along with your glucose. So here is an area, if I can come up with a novel bug which can also, for example, produce cellulase, hydrolyze the cellulose into glucose and xylose, and then also the same microorganism can convert that my sugars into, let us say, ethanol. I can do everything in one step. That's what we attempted to do in IIT-Madras, which is funded by industry. A two-step process. What we do is the first step is pre-treatment. Second step uses a fungus and a yeast co-fermentation. What the fungus does is to grow on the cellulose and produce the cellulose enzyme or xylanase enzyme, convert that into sugar, and then we have, for example, in the same vessel we have yeast cells also, right? The yeast cells which can ferment your sugar into ethanol. All you can do is one step. Or you can come up with the yeast, or you can come up with another organism which can have the cellulase gene, produce the cellulose enzyme and xylanase enzyme, and then also it has a fermentation capability to convert the sugars into ethanol. And we can develop new novel microorganisms for that particular application. So what we do is that in a single step, hydrolysis of biogroup and co-fermentation. But of course now in this we use two different organisms. One is the fungus, which produces the enzyme, which hydrolyzes my cellulose into simple sugars, and then the yeast to convert that sugar into ethanol. Okay, so what is the requirement will be that, but high yield requires fermenting all the biomass sugars, glucose, xylose, arabinose, mannose, and galactose. These organisms should be resistant to toxic materials and chemicals and hydrolyze it, because many of the process where we use some of the chemicals, harsh chemicals for pre-treatment, right? They can produce some toxic products. So even these small traces of these toxic products are present there in the liquid. These cells will not be able to function. But we need to develop a microorganism which can tolerate all these toxics. Should be able to outcompete the contaminants, because it's very difficult sometimes to sterilize these, right? If you want to have a protective fermentation, we need to sterilize all these raw material and then add your microorganism. But if you're going to have a process which is to be economical, we may not be able to sterilize that. We may not want to sterilize that. We may just hydrolyze that and then use open vats or at least semi-sterile vessels to convert your sugars into ethanol. So it should be robust in pH and temperature. Minimum metabolic byproducts you should produce. But one way is to develop these microorganisms by metabolic engineering. It can help in designing a tailor-made microorganism. Right? This is a new field, what is called metabolic engineering, where you can manipulate the pathway in a microorganism and then drive your sugars or your raw material towards your desired product. We'll see how we can do that in the. And I also pointed out there are possibilities other than producing ethanol from lignocellulosic material. You can produce a very large number of platform chemicals, right? For example, 3-hydroxypropylenic acid, or let us say xylitol, right? All you require is that you convert the cellulose into sugar and then use different microorganisms or different products to produce this biochemical, right? Maybe I can use one yeast to produce 3-hydroxy, or bacteria to produce 3-hydroxypropylenic acid, or maybe use some other yeast to produce xylitol, right? But one can produce variety of them. These are the potential platform chemicals, right? People are looking in industry to develop by the fermentation route. Of course, now all of them can be produced by a chemical route using petroleum as a crude as a source of raw material. But as I said, that we want to move away from the petroleum source. We want to go to renewable resources. So one can see how to develop. For example, succinic acid is a very important platform chemical. One can develop a biotechnological process. Then from there, we can go into many other one. Give an idea, for example, if I can produce 3-hydroxypropylenic acid, I can go into 1,3-propanediol, I can go to acrylamide, I can produce acrylic acid. Of course, these are chemical routes as of now. Right? But then once you get this 3-hydroxypropylenic acid by conversion of your lignocellulosic material by a fermentation route, right? For example, Dow Chemical, they have manipulated E. coli, right, cell pathway, manipulating them on at least about 10 different points in the pathway where they convert sugar into 3-hydroxypropylenic acid. They're able to get a very high concentration of this product and then recover this product from the fermentation broth, liquid, and then later on use a chemical route to convert them into various ones. So you can see that these are the important chemicals. Maybe in the next few years you'll find that they are now produced not by a chemical route, by means of biotechnological methods. Okay, other things that I think I'm running short of time maybe. But the other one that is in the area of biopolymers, which we are also looking at in IIT-Madras. One of them is called hyaluronic acid, which is to produce, it has got a lot of medical applications in eye surgery, in the treatment of knee joint, where, for example, people have arthritis. And if you can inject this hyaluronic acid in the knee joint, it lubricates and reduces that pain. So, but as of now, what is produced is produced from rooster comb. Right? But then people don't want to produce from the rooster comb because there is a possibility of contamination of virus from the animal source jumping into the human system. But there are many microorganisms which can produce that. So this is the property of hyaluronic acid. It's a very long chain molecular weight compound. It's a linear one, but you can see that the molecular weight of this compound is 10 million Daltons, as high as that. So, of course, there is a lot of applications in cosmetic industries because it can retain moisture. If you have low molecular weight hyaluronic acid, maybe 1 million or 2 million Daltons, each can go for cosmetic application. And of course, people are also using that as a health supplement. People take hyaluronic acid as a health supplement in the form of capsule because our body contains hyaluronic acid. We have maybe about 60 or 80 grams of hyaluronic acid which are present there in our body. But the cost of this medical application one costs about $100 per gram. So as I said that as of now, for these medical applications which are produced from the rooster comb, which is extracted from that, the comb of the rooster by a chemical method. Of course, you can also get from the bovine brain, but then of course there are many tumors and sarcoma and other cells also contain this hyaluronic acid. But what we do is now we can also produce this product by using a microbial method, right? Streptococcus pseudopremicus, or for example, L. lactis, right, can be used as a microorganism and allow these microorganisms to produce this product. Because we don't want to depend upon, as I said, an animal source for its production, especially if you're going to use it for medical application. If you're going to use it for cosmetics, it doesn't matter. But if you're going to inject this hyaluronic acid to the human body, and it's from the animal source, there's always a possibility of contamination with the virus. I mean, Also, you need to purify them to get this for medical application. You can see these hyaluronic acids produced with the microorganism as a capsular material. Okay. These are the cells, they are the protective layer of this hyaluronic acid. Why these cells produce the hyaluronic acid is because when they invade a foreign organism or foreign system, they want to protect themselves against the immunity proteins, right? So that they are not attacked by those immunity proteins and these bacteria or these are all destroyed. So they have some kind of a shield, right, protecting them. So those molecules which the body releases to protect against this foreign bacteria entering into the system cannot penetrate through this hyaluronic acid and attack the cell. So it's a defensive mechanism by the microorganism. But what we can do is that we can ferment these cells in the bioreactor, and then you can extract out this product. And as I said, the Streptococcus is an organism which produces that, but they are also pathogenic, right? They also can produce endotoxins, and which again we need to clean it up in the final product. But what we have done is to use, for example, Lactobacillus lactis. Lactobacillus lactis is a grass organism that means generally regarded as safe. And we have this organism in food industry in making curd and so on. So what we have done is we have metabolically engineered these cells to produce this hyaluronic acid. As such, L-lactis doesn't produce it, but the pathway for L-lactis and Streptococcus is almost similar to one another. So we could manipulate the pathway of the L-lactis so that the L-lactis is also able to produce this